A shark choked, not by another predator, by slime. Not metaphor, not exaggeration. A functional defense mechanism that expands 10,000 times its original volume in 4/10 of a second, faster than a human blink, and fills a predator's gill slits until the animal can no longer breathe. The creature responsible has no jaw, no spine, no paired fins, no stomach. By every structural definition biology uses to classify vertebrates, the hagfish, Myxine glutinosa, should not exist as a successful animal. It predates true vertebrates entirely. It is a craniate chordate, a lineage so ancient it belongs to a category of its own. The 13th of July 2017, Highway 101, near Depoe Bay, Oregon. A flatbed truck carrying 7,500 pounds of live hagfish overturned on a coastal 2-lane road. The slime that followed coated 11 cars, a state police cruiser, and a stretch of highway long enough that workers with squeegees spent hours pushing a substance that stretched rather than broke, pulling back toward itself like something alive. That was an accident. What the hagfish does to a shark is deliberate. Douglas Fudge, a biomechanics researcher at the University of Guelph, documented the deployment event in a 2005 paper published in the Journal of Experimental Biology. His team recorded the trigger-to-expansion timeline, 4/10 of a second. Fudge later described the volume. A single hagfish can fill a 5-gallon bucket with slime in a matter of minutes. The slime is not mucus. Researchers corrected that first. Inside the hagfish's skin, thousands of specialized cells hold the material in a collapsed state, threads coiled tight, waiting. The mechanism that causes them to unravel remains, as of the last published literature, incompletely understood. That is the question this video is built around. Not what the slime does, what the slime is. By the end, a specific number, measured in a university biomechanics lab and published in peer-reviewed literature, will reframe what a defense mechanism can actually be. The answer is not in the slime itself, but in what the slime is made of. Here is the obvious explanation. Hagfish produce mucus. Fish produce mucus. Every fish you have ever held leaves a slick coat on your hands. Same thing, right? Wrong. Pull a trout from a tank. Its body carries a thin, uniform film, a polysaccharide gel, simple in structure, passive in function. Drop that fish back in the water, and the coating dissolves within seconds. No threads, no architecture. Nothing left behind. That is mucus. That is what every biology textbook means when it says mucus. Hagfish slime is not that. Douglas Fudge at the University of Guelph spent years pulling apart exactly what hagfish slime is made of. His team's findings, published in the Journal of Experimental Biology in October 2005, were unambiguous. The slime threads are intermediate filaments made of proteins similar to those in human hair and skin, but with mechanical properties that exceed synthetic materials. Hair and skin, not mucus. The slime is actually two components, stored separately inside the hagfish's body. Mucin vesicles sit in one type of cell, protein thread coils in another. Neither component alone does anything remarkable. But the moment both hit seawater simultaneously, they self-assemble, threads uncoil. Mucin bridges them. A structural lattice forms in under half a second. This is not secretion. This is construction. Fudge's team clocked the full deployment at 0.4 seconds. No passive gel achieves that. No passive gel builds a lattice at all. A single fiber, pulled from that lattice with forceps, stretches to 3 times its resting length before holding. It does not snap, it absorbs the force and holds its shape. The "it's just mucus" answer collapses completely. The material self-assembles. The fibers are structural. The deployment speed has no biological parallel. But Fudge's data could not immediately explain one thing. If the two components are stored separately inside the hagfish, in different cells, never touching, what is the molecular trigger that fires them both at exactly the same instant? Here is the mechanism, not the effect, the mechanism. Inside each hagfish slime gland sits a single specialized cell, a thread cell, no larger than a grain of sand. Inside that cell, a protein fiber sits coiled under tension, waiting, not dissolved, not liquid, physically wound like a spring someone forgot to release. Contact with seawater changes everything. Osmotic pressure ruptures the cell wall in milliseconds. The coil explodes outward. The fiber unspools in a visible arc and locks into the surrounding mucin network, forming not slime, but a hydrogel lattice, structured, load-bearing, mechanically coherent. Douglas Fudge at the University of Guelph measured what that fiber can actually do. His team published the numbers in the Journal of Experimental Biology, October 2005. A single thread, 0.002 millimeters in diameter, registers tensile strength at 0.18 gigapascals. That is comparable to nylon at 1/50th the diameter. One fiber, thinner than anything you can see, stronger than engineered synthetic material. John Goslin at the University of British Columbia had already run comparative tests against spider silk. Spider silk, the material engineers have chased for decades. The hagfish thread held its own. A creature with no jaw, no spine, and no eyes was producing structural fiber that materials science had not matched. The 0.4-second deployment is not a chemical reaction, no enzyme cascade, no metabolic trigger, a mechanical spring release at the cellular level. The cell wall is the safety. Seawater pulls the pin. That changes the question entirely. Chapter 2 asked why the slime worked. The answer was architecture. But architecture implies design, and design implies a ceiling, some point where the structure maxes out. Fudge's data showed the ceiling had not been found. Scaling the fiber, increasing volume, increasing thread count, the strength scaled with it, linearly. So if a single thread already outperforms nylon, what happens when you collect enough of them to build something? And who, exactly, has already asked that question with a federal budget behind it? Here is the payoff. A single hagfish slime thread, thinner than a human hair, has a toughness-to-weight ratio that outperforms Kevlar. Not comparable to Kevlar, not approaching Kevlar, outperforms it. Douglas Fudge's team at the University of Guelph measured this directly, published in the Journal of Experimental Biology in 2005. The number held. The US Naval Research Laboratory took notice. Atsuko Negishi led formal investigation into hagfish slime proteins as a candidate material for next-generation flexible body armor, not a fringe idea. A funded, peer-reviewed military materials research program. Fudge et al. 2010, Ewalt et al. 2011, both documented, both traceable. This is what the slime actually is, not mucus, a protein fiber composite. Each thread self-assembles from a collapsed coil into a structural lattice in 0.4 seconds. The same 0.4 seconds from Chapter 1, the same Oregon Highway. The same choking shark. One mechanism, 3 demonstrations. But nobody has solved the coiling. The fiber requires a living cell to produce it. The coil structure, the internal architecture that gives it toughness, collapses the moment you try to replicate it synthetically. Researchers have extruded the protein. The thread forms, smooth, uniform, structurally hollow. The biological original is something else entirely. John Goslin at the University of British Columbia compared hagfish thread tensile properties to spider silk in 1999. Spider silk, already the benchmark for biological high-performance fiber. The hagfish thread was competitive. From an animal with no jaw, no spine, no eyes worth mentioning. The slime threads are intermediate filaments made of proteins similar to those in human hair and skin, Fudge wrote, but with mechanical properties that exceed synthetic materials. Read that slowly. Your hair, scaled differently. Deployed faster than a camera shutter. The Oregon spill in 2017, 7,500 pounds across Highway 101, was not a disaster. It was a field demonstration. Firefighters hosing elastic strands off car hoods were watching the same material naval researchers were trying to build in a lab. Neither group could fully explain it. The molecular trigger that initiates deployment remains unconfirmed. The exact uncoiling mechanism, the millisecond physics of a thread going from collapsed to structural, is still open in the literature. 300 million years of refinement, and the design is still ahead of us. That is the receipt from Chapter 1. The strange fact was never the volume of slime or the shark or the highway. It was always the fiber, a single thread, a number on a results table. A pencil underlining it in a university lab. The animal is still in its tank, motionless, coiled against the glass, waiting. If you found this fascinating, subscribe. We investigate strange, real creatures like this every single day.